Hello kids, today we will be discussing your Unit 2 of your Grade 7 Science, which is Properties of Material, in which we will be discussing things related to solution making and dissolving things, right? So the first subtopic of this Unit 2 is dissolving. Let's get into it. So what do we mean by the word dissolution or dissolving? So I will write two of these words, right, which are synonyms, right? So, dissolving, to dissolve something, or dissolution, right? The process of dissolving something, right? Dissolve means if you put, let's just say, some salt into water, right? So that salt would get, after a while you would find that the salt is gone, and if it were a colored salt you would find that the color of the solution or the water is going to be different. So whenever you take a solute, any solid or a liquid in that matter, when you put it in a solvent, it gives you a solution if they're mixed together, if they can completely become one, right? So what is a solute? Solute is always lesser in quantity, and solvent is always greater in quantity, right? And solution is the mix of both, right? And when you see something like this, in which there's like diffusion, right, going around, so this happens through diffusion, that when these salt is added into water, the salt particles break out into smaller sub particles, and they start diffuse all over the water, giving it a change of color if it's a colored salt, or diffuse it out, right? So what is diffusion? How the salt is moving. Let's just say if there is a dye, we have done this experiment in the lab, right? There is a dye. I put one droplet of dye into a water solution, right? A beaker filled with water. You would find that the dye colors is spreading evenly all across this beaker, right? Now, when we started this dye test, right, we saw that the dye color was slowly moving from one point to other points of the beaker, and after a while, when you guys went into the lab next day, you saw that the color was very, very uniform. This is when we call it's an equilibrium state, right? Now, the particles move through the liquid to diffusion, right? So what is diffusion? We have discussed this in our respiration chapter, that diffusion is movement of particles, right? When the particles move from high concentration towards lower concentration. And what that does mean is that if you look over here, then this red dye is in higher concentration. That means more particles are closer to one another, and there are no red particles over here, right? So it will try to diffuse into all other places, right? This is when we call it will start to diffuse. You will find that this dye is now spreading, and the color of the dye is becoming dimmer, and the solution is going to be evenly distributed, right? So this is when there is even distribution, right? We call it uh, equilibrium state, right? When the diffusion is consistently equalized in all places. This is also called homogeneous, right? Equal distribution of particles all over, right? So this is diffusion, right? I'm just going to rush through these topics, so I won't be detailing much, right? You can find some of the details in the book. I'm just going to give you a revision of the basic concepts which are going to be there. When we're talking about solution making, we understand that there are three types of solutions we can make, right? Number one is the unsaturated solution, in which if you take water and add a little amount of salt, you would find that all of the salt is gone, right? When all of the salt or the solute is gone, solute is the smaller quantity, when you add into the solvent and it's gone, it means it's unsaturated, and it can take more amount of salt before it becomes saturated, right? And what saturated means, that if you add more quantity of salt into a solution and it is unable to dissolve it, right, the salt is just going to sit at the bottom as insoluble, right? It is not able to dissolve. Then we call the solution as saturated, or when we say that the concentration is as much that it cannot take more solute, right? And supersaturated is when it becomes unstable. You heat it up for a while, you dissolve this extra salt, and when you let go the heating away, right, and the solution is going to cool down, right, this is at high temperature, right, higher temperature. If you put it to higher temperature, this extra salt will get dissolved temporarily, and once you remove this high temperature, the salt is going to crystallize at the bottom as becomes an unstable crystal form. So you will find irregular shapes of the crystals forming at the bottom, which we had seen in the lab, right? Now, what are the two factors that affect solubility, right? We had discussed several factors, so I'm just going to discuss these three main factors, in which two are very important. Number one is the temperature, because the more the temperature, more energy, right? Temperature means energy of particles, and definitely when the particles are more energetic, they're able to move much freely, they're able to dissolve or solve anything within themselves, right? So if it's a solute, it will dissolve quickly. If it's a solvent, even then higher temperature solvent will dissolve the salt quickly as well, right? So if you increase the temperature of the whole solution, the solution solubility rate is going to be a lot more fast, right? We see this that sugar takes very long time to dissolve in cold water, but if we just heat up the water a little bit, then you can easily dissolve sugar in water, right? You can try this, as a safe practice in home, in the supervision of your parents, if you want to try this, that how the solubility is affected by temperature. So do try sugar and water, and do not try salt, because salt is readily soluble in water, so you won't be able to differentiate much. But with sugar, it is going to be a very very clear view that even one spoon in one cup takes a lot of time to dissolve at room temperature, and if you just heat it up a little bit, you'll be able to dissolve that much quicker, right? Now, in terms of appearance, I told you that I'll discuss there are several types of solutions, right, in which when there is clarity in the solution, when this is what we call as homogeneous. Homogeneous means it's clear, and it is going to have same properties all over the place. So if I take a sample from here or here or here or here, the sample is going to be same properties, right? It is going to have same properties all over, irrespective of location. But compared to this, if I go for this one, if I take a sample from here, 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 and from the bottom, we will have different uh, properties, or we can say concentration, right, as well, because you can clearly see that at the top there's much clearer water or the solvent, and the middle there's some turbulence, some particles which are suspended, and at the bottom we see many particles sitting around. This is also known as suspension, right, or a turbid solution. So over here, as per appearance, we can define these mixtures of products as either solutions, which are completely soluble into one another. We have suspensions, in which some of the particles are suspended in the solvent, and some is excess is sitting at the bottom. And then we have the supernates, or the precipitate, in which supernate is a solution which is remained after chemical reaction, and it leaves solid particles, heavier solid particles at the bottom, which we call as precipitates, right? So these are some of the ideas regarding solubility, right? So if I try to draw solubility curves, right, if I want to investigate the solubility of any material, let's just say I have I'm just going to tabulate some of these values. So if we have salt, right, and solvent, or this what we call as solute, right, and it can be in grams if it's solid, or it can be in milliliters if it is liquid, but the solvent will always be in milliliters because it's a liquid that is trying to dissolve, right? So let's just say how it works. So we've got 5 grams, right? I took 5 grams, then I took 10 grams, then I took 15 grams, and the solvent that I'm using is 100 centimeter cube, right? So for all three, I use the same amount of uh, solvent. As you can see, in this case, if the amount of solvent and the solute, right, 5, 10, and 15. Now this is, let's just say, 100. You'd find that these solutions are able to dissolve uh, like this, right? Sorry. So you would find that as I'm going increasing the amount of solute, the concentration is increasing, right? So in other words, there are more solute particles present. So this is going up, right, in this axis. And when we let's not just plot this for now, right? Okay. Now just look from the numbers alone, right? If this is increasing, right, we're increasing the amount of salt, but if we have a beaker, so we had just 5 grams, right, over here. Now for this, we have got 10 grams, and for this, we now have 15 grams. So obviously, the one with the 15, right, is what we call as more concentrated, right? That means it contains more amount of solute in the given amount of solvent. For all three to be compared, we needed the same amount of solvent, right? But when we talk about the rate of solubility, then it does depend on how much solvent is there. So let's just say if we had these x and y axis in which we had solvent, quantity of solvent, right? This is solute and this is solvent. So if I plot this, so from 0, the more solute I add, the more solvent I'm going to need, up to a point that it is going to become straight, right? At this point, what we call as we have achieved a saturated solution, right? So it won't be able to dissolve much more amount of solute. Now if I try to dissolve the unsaturated part, so this part over here is unsaturated, right? Why? Because it is able to dissolve more amount of solvent, as you can see from these green lines, that it is able, sorry, green, yeah, right, it is able to take more amount of these as per the solvent is increased, right? I'm increasing solvent as well, but after a while, if I don't increase the solvent, right, it won't be able to dissolve much solute, right? That is what we call saturated solution, right? And if I draw a super saturated solution, and it is going to be, let's just say, if I plot it against temperature now, temperature and solute, right? So with the increase in temperature, we find that uh, the the solute or the salt is first rapidly increases, and then after a while it also stops. It won't be able to take any more of the salt, just salt will sit at the bottom, right? So increase in temperature also increases the ability of the solute to get dissolved. This what we can plot as solubility as well, right? The rate of solubility, right? A little brief of how the solubility works, that the more temperature is increased, the more solubility of the particle or the salt is going to increase. Now over here I've plotted solutes, not the solubility, right? This is the solubility curve, a little bit different from the ones that I'm drawing, which involves only the quantity of solute or the solvent, or solute versus temperature. We're not talking about solubility directly, right? We're just representing that or relating it to it, right? And now finally we discussed paper chromatography, in which it is a separation technique, right? So paper chromatography is a separation technique, right? Sorry. And what it does, it separates solutes or dyes from a mixture depending on solubility of each component. And when we were doing this experiment in the lab, I told you that everyone was given the same sample, but you saw that some of the components went a little ahead and some components stopped earlier, right? And why this is happening? Because each of these components had a different rate of solubility. This is the solvent which rised up in the on this paper. This paper is what we call as chromatogram, right? So on the chromatogram, we when we placed our dye, our sample, right? This is a sample. when the solvent went up, it got absorbed. Then when the water moved up, it separated these colors dyes, right? And each of these dye had a different solubility rate at which it could dissolve in water. Hence, it moved to a specific distance before it stopped going up. As you can see, the red is traveling the most, and even in our experiment, we saw that the blue and the orange had stopped earlier, but the red was the last color that we were able to see on the highest level of this chromatogram, right? So we did see that the blue had streaks initially, but afterwards orange took over. We didn't had the yellow dye, but we did had this black, blue, orange, and red dye, right? So we tested on these four colors, and we were able to separate these, right? Rest, I did give complete instructions on how this technique works and what is the process of doing this technique. So just to give you a brief idea of what really meant, what it really means to do chromatography, it's just a separation technique which helps you separate solutes from a mixture of solutes from one another. Each component, if you want to separate the components. This is not to identify the solvent, but the components in each of these samples, right? This sample could be containing 5, 6, or 10 samples. It's a mixture. So if I want to see how many components are there in this one, so it is basically what I will call as qualitative test, right? So it can only give you at least at your level, we're going to consider this as a qualitative test. Qualitative test means it can only identify, right? It will only identify components, and it will not tell you any results, right? So no results, right? You only get conclusion in this, right? Whether there are how many components, whether there are 5 components, say there are 6 or just 2, and which one is more soluble and which is not, still you cannot calculate those things, at least at your level. So it is just a qualitative test for you guys. But in IGCSE, we're actually going to study this as a quantitative test, where we can calculate some of these solubility rates as well, right? But at your level for now, in grade 8th, we're just considering this as a qualitative test. I hope I was able to give you some of the feedback. Some of your questions which are still there in your head are now clarified. If you still have any questions, just post a comment or just text me up. I'll try to respond as soon as possible. Thank you.